Good morning, everyone. All right. We will continue looking at some of the functions of our biologic membrane. I expect that we will be able to finish that in a timely manner today, and I will start looking at our next chapter of material. Chapter 3, we'll start looking at our energy resources and energy processing in cells. I hope that some people who have purchased the SmartWorks, that our e-textbook, and the SmartWorks that comes along with it, had a chance to look at the hair cells videos. I would recommend today we'll be looking at the action potential again and propagation of that action potential. They have a nice animation of that, as well as an animation of the sodium potassium pump that we'll talk about today as well. So everyone who's here, let's make sure that you've had a chance to log into your Reef account and you have joined our course. Everybody good? All right. So where we left off last week, we had discussed the different phases of the action potential that a traveling wave of the electric impulse, the changes in our membrane potential. I see a hand raised, yes? Is there a reason? Camilo has your hand raised. Do you have a question for me? Is that a mistake? Can you put your hand down? Okay. Can everybody hear me okay?
Yes, Dr. Bear, we can hear you.
Okay, fantastic, thank you. All right, so here we are looking at an axon, and what we're going to look at next is how those action potentials can travel in one direction down that axon. This is the nerve impulse, the traveling wave of these action potentials. We remember that the changes in the voltage across the membrane will affect the shapes or the conformation of our voltage-gated sodium ion channel. When that membrane is polarized, when the cell is at rest, we have a higher concentration of negative charges on the inside of that cell that are attracted to the higher concentration of the positive charges outside the cell, sort of holding a thin film of those charges on either side. And that sodium ion channel is in its closed conformation at that membrane potential, and we call it polarized. A stimulus that causes the membrane to become less polarized will cause a change to that ion channel such that it will open. Right? The membrane potential changes from -80 to about -60. At that state, that channel, that protein will change its shape and will open, which will allow positively charged sodium ions to enter the cell, because we know that all cells have a higher concentration of sodium ions on the outside than they do on the inside. And we'll see why that is. That is due to the sodium potassium pump that's constantly pumping sodium ions out of the cell, a different protein in the membrane. So once the cell is converted from its closed state to its open state, the continued influx of positively charged sodium ions will continue to depolarize that membrane such that there will be a big change, many more positive charges on the inside. The membrane potential rises into the positive range, which will cause that channel to convert to yet a third state, a third shape in which it's inactivated. It's not only closed, not only does sodium not pass through, but it's inactivated. It can't be converted back to an open state. It will have to wait until the membrane is repolarized. The membrane will then be, the membrane potential will then be changed back to this polarized state. The channel will then convert to its closed state. Remember, it's only in this direction. The channel has to be closed before it can be opened. It's open for a very short period of time, becomes inactivated, and then will go to the closed state. And we're going to see these changes, which are going to be part of the perpetuation or the propagation of these changes in membrane potential. So here we're looking at an axon, and we're going to look at how that action potential moves along an axon, essentially as a wave of depolarized region of a membrane, moving in this case from the left to the right. So just to orient ourselves here, here is our axon, and we have many sodium ion, gated sodium ion channels in the membrane, right? Looking at the top, looking at the bottom, essentially this is a longitudinal section through an axon. And all of those sodium ion channels are in the closed state because our membrane is polarized. Right? We're looking down here, resting, our membrane is polarized, more negative charges inside compared to the more positive charges outside. At this portion of the axon, we see in blue this depolarized region of the membrane where there are more positive charges inside than there are outside. That depolarization will cause one of the, or a set of the sodium ion channels to become open, right? So closed state when at the resting or polarized state of the membrane, where the membrane is depolarized and we're moving from minus 80 millivolts to about minus 60 millivolts, causes the sodium ion channel to pop open. And when it opens, if we follow our orange arrows, that will allow sodium ions, which are higher concentration outside the cell, allow sodium ions to enter. That will further depolarize this region, which means that the sodium ion channels here that we're looking at that had previously been open will now convert to their inactivated state, right? All within this region of depolarization. And those channels will remain inactivated until the membrane becomes repolarized, when they will convert to their closed state. Now, what we're seeing here is that the propagation, this continuous wave of this depolarized region of a membrane is going to move in one direction. In our case here, within a millisecond, it's going to be moving to the right. And the next sodium ion channel that are closed at this point will be opened, because the opening of this channel will depolarize the membrane, which will result in the next sodium ion channel, which is currently closed. A millisecond later, this sodium ion channel will be open. So our next slide is we're going to move in time, a millisecond in time, and notice our depolarized region in blue will be moving to the right, meaning that the sodium ion channels that had been open will now be in the inactivated state. The sodium ion channels that had been inactivated will be in the closed state. The sodium ion channel that had previously been closed will be in the open state, right? Because sodium ions had been diffusing into the cell from the neighboring sodium ion channel that had previously been open. All right, so we have this wave, and the previously open sodium ion channel and the movement of those ions are the stimulus that cause the next closed sodium ion channel to pop open. Again, this wave is only going to move in one direction, only move to the right, because the sodium ion channels that had previously been opened can only move to an inactivated state, and they will temporarily be inactivated until the membrane is repolarized. And then they're closed, and they can then be stimulated by the next change in membrane potential that will allow them to open again. So we can have a traveling wave pass down, another wave can pass down, but it can only move in one direction. Now, that's a little hard to be able to see looking at just a couple of static images. And so this is where I think it would be a great idea to go take a look at, in SmartWork, the action potential, where they have, they will actually reiterate the changes in membrane potential and the associated changes in sodium ion channels that we discussed, showed in an animation. But I will, excuse me, I was going to actually show that very quickly here. Let's see if I can, oops, let's go to it. And if I, without showing the whole thing, because we don't need to see it all, I thought I could make this larger. There we go. Okay, hold on a second. We want to start in the middle here. Hold on. I want to start a little bit later on. Right there. There we go. So if you notice, what you're watching is the changes in the membrane potential, and those changes in the membrane potential are changing the shape of the sodium ion channels. And the changes in the sodium ion channels are causing a subsequent change in the membrane potential. Right? And the directionality of the ability of those sodium ion channels to change, right? Only from closed to open, have to be inactivated, wait until the cell returns to, or the membrane returns to, the polarized state when it can be closed again. Now, one question that we might be concerned about is, well, how is it that our membrane, even in a resting state, is polarized? And how is it that, in order for all of these membrane potential changes to occur, when a sodium ion channel is open, how is it that the sodium ions are able to enter the cell and then change the membrane potential? And that is because the cell is going to maintain a sodium ion concentration gradient across the membrane. That's an essential feature of a cell being alive, is having this concentration gradient of these ions. So how does that occur? How is this resting membrane potential established, and how, which really means, how is it that the cell can maintain concentration gradients of different ions? How is it that our membranes are polarized. We have a higher concentration of positive ions outside, lower concentration inside, and specifically essential for this electrical impulse is a higher concentration of sodium ions outside the cell. What we're going to see is that a pump, a membrane protein, carrier protein, can actively move sodium from the inside of the cell to the outside. And at the same time, it will move potassium ions from outside the cell to the inside. This is active transport. Movement of ions against their concentration gradient requires ATP. Our cells that have these sodium potassium pumps consume about 30% of all of the ATP that's utilized by cells. And in our brains, about 20% of all of the ATP used in our brain cells, our neurons of our brain cells, are used to drive this sodium potassium pump. So what we're going to see is, first of all, we'll spend some time looking at the action of the sodium potassium ion pump, and then we will discuss how that impacts the resting membrane potential. Because if we don't have a resting membrane potential that is more negative charges inside, more positive charges outside, then we can't have any changes in the membrane potential, which means that there wouldn't be any electrical signal moving along an axon. So here is a diagram of our sodium potassium pump, this purple protein, transmembrane protein sitting in our lipid bilayer. Here we have our resting membrane potential, more negative charges on the inside of the membrane, more positive charges on the outside of the membrane, and reminding ourselves that we have these two opposing ion gradients. In addition to, not only is the sodium ion gradient different from the potassium ion gradient, that sodium ion gradient is a very steep gradient. We have about anywhere from 10 to 30 times more sodium ions outside the cell than inside, and this is going to be cell dependent, cell type dependent, much higher concentration of sodium ions outside the cell due to the pumping of sodium ions from the inside to the outside, which means that an ion channel, a sodium ion channel, when it is open, will be able to allow very rapid influx of sodium ions, right, from the outside of the cell to the inside. So we have this very steep chemical gradient of sodium ions because we have a big concentration gradient, but we also have this electrical gradient where it's very easy for a positively charged ion to be able to pass through its channel from the positive side to the negative side. We have the opposite case in potassium ions, larger concentration of potassium ions inside, partly due to the fact that potassium is going to be pumped from the outside to the inside via our sodium potassium pump. But also we have this very small movement of potassium ions out of the cell because the electric gradient is opposite to the chemical gradient. It's hard for a positively charged ion to be able to move out of the cell from the negatively charged side to the positively charged side. So we're going to take a look at our pump, this protein in the membrane that will actively transport these ions to opposite sides via the hydrolysis of ATP. So ATP, the energy resource, is absolutely required for this pump to be able to function. How the pump works is that it will bind 3 sodium ions on this cytoplasmic side of the protein, which will cause a change to the shape of the protein that will activate its ATP activity. This sodium potassium pump is actually an ATPase, meaning it will cleave ATP, hydrolyze ATP, and transfer a phosphate group from ATP to itself, which causes another change in its shape. That shape change will then allow two potassium ions to bind from the extracellular side. That binding causes another shape change that results in dephosphorylation, the removal of that phosphate group, which causes yet another shape change. And we can see all of that on our next slide here. So a simplified view of our sodium-potassium pump sitting in our lipid bilayer, multiple conformations to the three-dimensional shape of that protein. So our pump here in green, in the starting, so you notice we have a cycle of these different changes. The protein actually has 3 binding sites for sodium ions. We're only going to follow one at a time. 3 sodium ions will bind when the protein is in this shape. The binding of those sodium ions will allow the protein to be able to hydrolyze ATP, convert ATP to ADP by transferring that terminal phosphate from ATP to the protein itself. Right, so it will phosphorylate itself. When the protein phosphorylates itself, it causes a change in the shape of the protein. That change in the shape of the protein will allow the sodium ions to be released into the extracellular space of the cell. The reason for that is the change in shape causes those sodium ion binding sites to have less affinity for the sodium ions, so they're released. But that change in shape also exposes some high affinity potassium ions. Therefore, and there's 2 of those sites, we only see one here, those 2 potassium ion binding sites will allow binding of 2 potassium ions. The binding of those ions cause a change to the shape of this protein, which will release the phosphate. Once that phosphate group is released, that results in a change of the pump again to its original conformation, which causes a change to the potassium binding site so that it is now low affinity, lets go of the potassium ions. They have now been transported into the cell, and then exposes the high affinity sodium ion binding sites again, allowing the cycle to begin. So these binding of ions causing phosphorylation, causing a change in shape, resulting in release of sodium ions to the outside, picking up potassium ions, causing a change, releasing the phosphate again. This cycle of this pump requiring the availability of ATP to help drive this cycle will result in pumping 3 sodium ions out of the cell for every 2 potassium ions it pumps into the cell. That's why we call this active transport, requires energy, and it is pumping, it's moving these ions against their concentration gradient. So I have a question for you here. This pump is maintaining the high sodium ion concentration outside the cell, the high intracellular potassium ion concentration. So in that cycle that we just described, the changes in shape, the phosphorylation and dephosphorylation events, what we're asking here is what happens to this pump, or what happens in this cycle right after the pump hydrolyzes ATP. As long as ATP is available, the pump will hydrolyze it. So the pump actually has intrinsic ATPase activity. How are we doing? Everybody had a chance to vote? All right, let me close it up. See, one last vote came in. All right, I'm closing it up. And yes, D is the correct answer. When the pump hydrolyzes the ATP, it will phosphorylate itself. It will transfer a phosphate from ATP to the pump, which causes that pump to change its conformation, change its shape, which allows the release of those bound sodium ions. So in looking at the action of our sodium potassium pump, all we've really done so far is describe how it is that these ions do have a gradient across the membrane, right? Why we do have a high concentration of sodium ions outside and a high concentration of potassium ions inside. But it doesn't really explain why our resting membrane potential is in the minus range, right? More negative charges inside the cell than outside. And the reason for that is that not only do our membranes have a sodium potassium pump that is pumping sodium out and pumping potassium in to create these gradients, our membranes also have gated sodium ion channels allowing sodium to diffuse back in. Our membranes also have potassium ion channels that will allow potassium ions to flow out, right, down their concentration gradient. And what we're looking at here is that while our cells do have a potassium ion channel that will allow potassium ions to move down their concentration gradient from inside to outside, if our cells did not, or if that potassium ion channel was closed, we would end up with a membrane potential of 0. If if potassium ions were not able to diffuse out of the cell, there would be an increase in positive charges inside the cell that would essentially match the positive charges outside the cell, and we would have a membrane potential of 0. There would be no voltage across the membrane. But in fact, our cells do have a potassium ion channel that is open. And this does allow these diamond-shaped potassium ions to be able to move down their concentration gradient, right? We have a force. We have the potential energy of the concentration difference that allows these ions to diffuse from inside to outside. However, that is in opposition to what we call an electromotive force, the force of the membrane potential, right? While potassium ions want to be able to diffuse out down their concentration gradient, the fact that it would leave behind negative charges means that there is a voltage across the membrane, more negative charges facing the inside of the membrane compared to the positive charges outside the membrane, which causes a balance, right? Prevents too many potassium ions from diffusing out. And so these opposing forces of concentration gradient driving potassium out, electromotive force preventing potassium ions from moving out, leads to an equilibrium, an electrochemical equilibrium. Every time a potassium ion tries to move out, another one is forced to remain in. And so we call this the potassium ion leak channel. That leaking potassium ions maintains this charge difference across the membrane so that our membranes do have a negative voltage across a living cell's membrane. Now, we can see how that would be important in cells that have electrical activity to have these arresting membrane potential that can be altered, right, can be depolarized, that can allow for movement of these ions. And as we just saw, these action potentials that are propagated down an axon. But any cell, any living cell, has a negative voltage across its plasma membrane. Different cell types will have different values to that voltage, the difference in charges across the membrane. But these ion gradients are essential to be able to maintain this negative voltage across the membrane. And these ion gradients are essential to counterbalance the large amount of molecules that are inside the cell and maintain a balance of solutes outside the cell and inside the cell to prevent water from constantly flowing into our cells, which would result in our cells swelling and eventually bursting. So the fact that our cells maintain not only this negative voltage across the membrane, but maintain a steep gradient of sodium ions such that there's a much higher concentration of sodium outside the cell than inside. In addition, there are other pumps that will actively transport negatively charged chloride ions outside the cell, which is why our cells are existing in biologic fluids that are very high, they're very salty, right? Very high in sodium ions and chloride ions. This maintenance of this high ion concentration outside the cells allows the cells to exist within an isotonic solution, maintaining an equilibrium of water movement for every time a water molecule diffuses into the cell, another water molecule will diffuse out of the cell, so that our cells don't shrink or our cells do not burst. We're maintaining the osmotic balance that allows a cell to be able to maintain its equilibrium with the solution that it's floating around in. In addition, these ion gradients are essential for different types of functions of cells, not only establishing electrical currents across the cell. In the case of electrical cells that can generate electricity, like our neurons, but other types of physiologic functions. For example, it's critical to have a high concentration of chloride ions outside the cell, right? There is a chloride ion transporter that will pump chloride ions outside of the cell. They, of course, will have a hard time diffusing back into the cell because they would be moving against the membrane potential, which is negatively charged inside the cell. And we know of examples of disorders that arise due to an imbalance in these ions across the membrane. And one example is the disorder cystic fibrosis. Cystic fibrosis is caused by a defect in a chloride ion transporter. That transporter can't move chloride ions out of the cell. And so that chloride ion gradient is disrupted. That will affect the osmotic balance of the cell. Now, these chloride ion transporters are essential features of the epithelial cells that line the respiratory tract, that line the digestive tract, in order for those cells to secrete products that are essential for functions of those of the tissues of those organ systems. And in the absence of a proper chloride ion gradient, the cells will be secreting too much salt. They will be secreting mucus that is very salty. They will be or too thick. They'll be secreting sweat that is too salty. They won't secrete pancreatic components like insulin and digestive enzymes properly. And so that will affect proper digestion functions of the intestine and will affect the lining of the respiratory tract. And so while there are different treatments for cystic fibrosis, some treatments that can break up the mucus, antibiotics that try to reduce the amount of bacterial infection in the bronchial tubes and in the lungs, because the bacteria find that sticky mucus secretions to be a really good medium for their growth. This inability to maintain the proper chloride ion gradient across the cells will frequently lead to fatality within middle years, 30s to 40s, for people who have cystic fibrosis. So if we look at our chloride transporter, we call it a CFTR, cystic fibrosis transmembrane conductance regulator, meaning a protein, a carrier protein in the membrane that will actively transport, because it will interact with ATP, or it requires ATP hydrolysis to drive chloride ions out of the cell. So defects, mutations in the gene that encodes this membrane protein result in its not folding properly, and the misfolded protein was not able to transport chloride ions properly outside of the cell. So yes, so the chloride ions will remain in the cell instead of being pumped out. So we don't have the higher, this disorder would result in the absence of a gradient, higher concentration of chloride ions outside than inside. So then the cell would have an osmotic imbalance, right? And the cell would not secrete its products properly. So, as I like to give you at the end of a chapter, a list of things that you should be considering while you're studying through this material. Thinking about, first of all, the structure of our biologic membranes. What are the roles of the lipid components, protein components, carbohydrate components? What's the roles of some of these transport molecules? What's the difference between passive transport and active transport? What we were just talking about today is these active transport carrier proteins that move ions against their concentration gradient. Again, thinking about function, how we spent a lot of time talking about how sound waves are transmitted to the auditory nerve. How does that involve ion channels? What is an action potential? How's it propagated along the axon? And why is it important that these cells generate, as we were talking about today, these ion gradients, differences in concentrations of different ions across the cell? Okay, so what I'd like to do is start our next chapter. And again, I highly recommend people who do have their smart work from our textbook that you've watched through those animations, because I think they're really valuable, really nice way of visualizing the kinds of changes that are occurring. So our next chapter, we're going backwards in our textbook, and we're going to introduce the concept of the energy resources in a cell. But really we'll be talking about in this chapter, just starting to think about some of these biochemical processes, is why do cells need energy, and how do cells use the energy that they acquire to create macromolecules, to create order in a cell. And so we want to just talk a little bit about the use of energy in a cell, right? Before we start talking about our energy generating organelles, we'll be talking about mitochondria, we'll be talking about chloroplasts that either acquire and or utilize energy for the cell. But we want to spend a little bit of time talking about spontaneous reactions, spontaneous and non-spontaneous reactions. And we need to introduce or remind ourselves on the concept of free energy in a cell. In any system, you can measure the energy of a system, but the energy that's relevant to a system being able to do something, for a cell to be able to do the work of being alive, you need to think about free energy. So free energy is the energy in a system that can do work, that can make something happen. And if we look at just one biochemical pathway in a cell, such as glycolysis, which we'll be talking about in our next chapter, the breakdown of glucose, which results in the cell being able to generate ATP, a mobile energy carrier within the cell. And if we look at the profile, the energy profile of this chemical reaction, what we see is free energy changes. Glucose is a high energy molecule. But if we look at the different steps of the chemical conversions of glycolysis in a cell, what we see is over the process of this entire biochemical pathway is that the overall free energy change is negative, right? The free energy of the cell changes, and mainly in three big steps. Loss of energy during the process of glycolysis. Two of these steps are actually due to the hydrolysis of ATP. One of the steps is due to the synthesis of ATP. So at each of these big steps, ATP is either consumed or it's generated. But either way, the amount of free energy in the cell is reduced. And that's due to the thermodynamic fact that while the first law of thermodynamics is that energy in the universe is constant, energy cannot be created or destroyed, but every energy conversion or every energy transformation or transfer results in the loss of some free energy, the conversion of some energy to the lowest form of energy, which is heat, which is dissipated, which is the most disordered form of energy. So if we turn to thinking about this in the context of a cell, every time an energy transfer occurs, some of that energy can be used to do work in the cell, to drive a chemical reaction, to cause a movement in the cell, but some of that energy is converted to dissipated heat. So if we think about a breakdown reaction in which, let's say, sugar is broken down like we just saw, some of that spontaneously, that overall spontaneous process would end up releasing heat, the most disordered form of energy. But cells don't always break down macromolecules. Cells have to create order within them, within that living unit. And so while a cell, it has a number of very small pieces of matter, the cell has to acquire energy in order to be able to build macromolecules within the cell. And even while the cell is creating order inside by creating structures and macromolecules, that process of building molecules is going to generate heat. In that process, some of the free energy is converted to the lowest form, the most disordered form of energy, which is heat. and that heat will be dissipated out of the cell, increase the disorder of the universe. So that second law of thermodynamics is that every energy transfer or transformation will lead to increased disorder of the universe. Even if there's more order locally, it will contribute to more disorder of the universe. So I'd like to ask you a question. A question about matter, not specifically about energy, but it's the same kind of principle. Thinking about taking in about a bunch of matter when you're eating, taking in your fuel, your food, your matter and your energy. If you take in 4 pounds of food during the day, does that mean that your body mass will be 4 pounds heavier the next day? Everybody have a chance to vote? Can I close it up? 5 more seconds. All right, let me close it up. And the answer is no, you will not weigh 4 more pounds the next day. And the reason for that is that while the food that you take in, only a small portion of those atoms and molecules will be incorporated into your cells, right? Into molecules in your cell. Along the way, even while digesting food, a lot of that matter will be released as carbon dioxide, will be released as smaller, more disordered forms of matter, like water and carbon dioxide that you exhale. Right? So you take in these complex molecules, but you end up releasing breakdown products, smaller, more disordered molecules. And that's the same thing with the energy. You take in high complex forms of energy, the chemical energy in the bonds holding together all of those food molecules. And some of that energy is transferred to the energy of motion, the energy of the cells doing the work. But a lot of the energy, in fact, most of the energy carried in the chemical bonds of those complex molecules that you have taken in as fuel, most of that energy is released as the most disordered form of energy, which is heat, right? Thermal energy, which will be dissipated from your cells, dissipated from your body, adding to the disorder of the universe. So we want to be thinking about this concept of contributing to disorder of the universe as as living cells, and think about how living cells are different from non-living things of the universe, and how that comports with the second law of thermodynamics. The classic example demonstrating disorder, the spontaneous conversion of ordered structure to disorder, is what happens to someone's bedroom. Spontaneous occurrence of disorder. Higher disorder, which is defined as entropy in the universe. So here we have a very old-fashioned, this looks from the 70s, ordered bedroom. Everything has a place, it has structure, it can do something, right? Highly structured, highly ordered. Over time, that room will spontaneously become disordered, right? This is energetically favored from a highly structured, but actually very unstable arrangement of all of these items, which will over time spontaneously become less ordered, less organized, more disordered, right? And we can think of anything that's non-living is going to become disordered. Buildings will crumble over time. Water at a high elevation will flow downhill to become more disordered. An organism that dies will decay. Right? So anything that's not alive will spontaneously become less ordered, will spontaneously contribute to the disorder of the universe. Now, in this system of this bedroom, this collection of matter can be more ordered, but the establishment of order would require the input of energy, right? It would require you running around, picking up everything, moving your muscles, sweating, contributing heat to the environment, or if you're vacuuming, right, applying electrical energy, a lot of that energy will be transferred to heat energy as well. So even when you or someone or a living cell is establishing order locally, it's always going to be contributing disorder to the universe. But this also demonstrates the difference between spontaneous reactions or spontaneous processes as the room becomes disordered. A non-spontaneous process is one that requires energy to be able to build or create order. So let's bring this to an actual cell and thinking about the chemicals, the compounds that are within a cell. If we have a reaction, Y is converted to X, what would make this reaction a spontaneous reaction? What could we say about Y compared to X that would make this chemical reaction spontaneous? Meaning that Y will be converted to X. Anyone give me a guess? X would have to have lower free energy, right? To make this reaction a spontaneous reaction, X would have to have less free energy than Y. Free energy is the amount of energy that can perform work, that can do something in the cell. Spontaneous reactions are reactions in which a compound or a molecule of higher free energy is converted to lower free energy, meaning that the energy of Y is released as Y is converted to X. We call that a spontaneous reaction. It's going to increase disorder of the universe. We can look at it this way. If we have the reaction Y to X, if the free energy of Y is greater than the free energy of X, the conversion of Y to X means that the delta G, G is free energy, is less than 0, it's negative. Disorder of the universe increases. This can occur spontaneously. We would say that that's an energetically favorable reaction. Of course, the opposite reaction could occur. X could be converted to Y, higher free energy, but that can only occur if energy was added to that reaction. Right? Conversion of X to Y, this energetically unfavorable reaction, would require a source of energy to help drive that. Right? It would have to be coupled to a second energetically favorable reaction to be able to drive an energetically unfavorable reaction. So that's the kind of things we're going to be talking about in this chapter. The difference between a spontaneous reaction and a non-spontaneous reaction, and how it is possible that a non-spontaneous reaction can be driven forward only if it's coupled to another energetically favorable reaction. So we'll end here because I have run out of time, and we will pick up with chapter 3 and continue talking about that the rest of this week. All right. So I have chat office hours today at 12:30 if anyone has any questions for me. Otherwise, I will see you Wednesday in class.